Hey folks, welcome to the show, and today we will be discussing protein synthesis and each of the steps that occur in the process, performed by Micah, Carter, Ari, and Wilson.
First of all, what is transcription?
Transcription is the process of copying a base sequence from DNA to RNA.
Transcription starts with RNA polymerase binds to a promoter, which is a region of DNA with a specific base sequence.
RNA polymerase then uses one strand of DNA as a template to make the complementary nucleotides in the strand of RNA. RNA polymerase only binds to promoters. Promoters are regions of DNA with specific base sequences. Before these RNA molecules can be read, they require editing, where bits are cut out, introns, and some pieces remain exons. Shown in the drawing, pre-RNA has a cap on the left and alternating parts of exons and introns with a tail on the right, which then zigzags where the introns are removed and thereafter is the spliced remaining exons.
To be able to quickly copy a DNA sequence into RNA allows for thousands of RNA molecules to be produced.
During transcription, a piece of DNA that codes for this specific gene is copied into mRNA, which is messenger RNA, in the nucleus of the cell.
The mRNA then carries the genetic information from the DNA to the cytoplasm, where translation occurs.
The next part of the process is involved in the making of a protein.
Proteins are made by the joining of amino acids in a chain called polypeptides. The specific order of amino acids in the polypeptide chain determines the shape, chemical properties, and the function of protein.
The four bases of RNA are A adenine, C cytosine, G guanine, and U uracil. These bases form a genetic code. The genetic code is read 3 bases at a time.
The 3 base word is called a codon. It consists of 3 bases that specify a single amino acid to be added to a polypeptide chain.
Now we are heading into translation.
Translation is the decoding of a mRNA message into a protein.
Translation begins when a ribosome attaches to an mRNA molecule in the cytoplasm. Each transfer RNA has an anticodon whose bases are complementary to bases of a codon on the mRNA strand.
As each codon passes through the ribosome, molecules of tRNA bring amino acids to the ribosome. Each tRNA molecule carries one kind of amino acid, and it has a 3 unpaired base called anticodons. These are complementary to codons.
The ribosome joins 2 amino acids together and breaks the bond between each tRNA and its amino acid. After tRNA leaves the ribosome, and the ribosome grabs the next tRNA until the polypeptide chain is complete. Then the mRNA is released from the ribosome. What happens if something goes wrong during the process of protein synthesis?
It results in a mutation. Mutations occur when an incorrect base is inserted or a base is skipped during transcription.
One example of a mutation is a mutation that can change a single base pair called point mutations. They occur at a single point in the DNA sequence.
For example, if a mutation changed one codon from mRNA from CCC to CCA, this is an example of a silent mutation because they don't affect the amino acid sequence.
But changing from CCC to ACC is more significant because mutations that change amino acids specified by a codon are called missense mutations.